Hello everyone, in this next lecture video we're going to begin to discuss the concept of what we refer to as enzymes. Alright, so let's get started. Alright, so in order to release chemical energy, so we were just talking about macromolecules and we said that energy is stored in the form of either lipids or carbohydrates. So if we want to release that energy that's stored in these macromolecules, what we actually got to do is start to have a way to easily break and release the energy that is stored in the chemical bonds between all of the monomer units. So if I start to break apart a polysaccharide, I'm going to start to release one glucose at a time. Well, the energy is going to be in those bonds as we start to break them down. All right, so enzymes are actually what help us do that, as well as almost every other particular process that occurs within our bodies. So let's begin. An enzyme is also what we call a biological catalyst. A catalyst is something that gets something going quickly and immediately. So right now, if you're at home and nice and calm and nothing was bothering you, but if your brother or sister walked up and smacked you in the face, you would probably instantly get really ticked off and start chasing them around the house. Well, guess what? They just catalyzed the reaction in you. They got you to do something quickly. That's what an enzyme is going to do. All right, so what an enzyme for us is going to be a specialized protein. So it's a protein-based catalyst. It's going to work in biological organisms. And it's going to speed up or start a chemical reaction. And the thing about it is, it doesn't get changed itself, which means it'll perform the reaction, and when it's done, it's left untouched. And it can go and do another reaction over and over and over again. So it's like a little Sonic the Hedgehog down here, going to be able to move and do things quickly and do them over and over and over again. Now, ultimately, what it's doing to help these reactions happen quicker is it's reducing the amount of what we call activation energy, which is the energy that's needed to start any reaction in your body. All right, and again, the enzymes are going to lower this amount. So you can think of activation energy as the amount of money needed to start a business. Let's say that a business is going to cost you $100,000 to begin. Well, you don't have $100,000. Maybe you have $20,000. Well, somebody, the bank, family, friends, anybody else you may know, let's say they go and loan you the $80,000 so you can now have $100,000 and start the business. That's what enzymes are doing. They're loaning out energy to make things begin to happen and happen quickly. And again, an enzyme is a protein. All right, remember we talked about protein structure. So up here you can see where I have my cursor, a little laser pointer. This would be a tertiary structure protein. You can see amino acids linked together. You see the little yellow lines representing hydrogen bonds, making it take on this structure. So proteins are actually going to look like this. All right, I'm sorry, enzymes are actually going to look like this. They're proteins, and again, they're made up of amino acids. Now, this diagram is what generally trips up a lot of students every year. So let's make sure that we take the time to understand this. So what we're looking at with this diagram is we're looking at two reactants, glucose and oxygen. These are the two items that we're going to use to produce energy. When we break them down, we end up with carbon dioxide and water. So we're actually looking at the process of cell respiration that's going on in our bodies right now. So all the energy is in this glucose molecule. We're going to just need oxygen to help us along. And as we begin to break the bonds in glucose, we release energy in the form of ATP, which isn't shown, but then we also have the two waste products, CO2 and water. So whenever you look at a graph like this, wherever the reactants are, and let's say right here at this dotted line, that's the starting point. All right, from this point, that's the potential energy line. So let's say 100 calories you can get out of this one glucose molecule, just for arbitrary sake. So when I break glucose and oxygen down into CO2 and water, my overall energy released during the reaction, in theory, is going to be 100 calories. However, that is not going to happen just spontaneously. All right, it's going to need some help in order to occur. If it did not have help, if the reaction here, the breakdown glucose, didn't have any help, it's going to require a much larger initial investment of energy to actually get this going. And that's what you see by this large red curve going up here. It says without enzyme. So that's the activation energy needed in order to get to this point right here where you start to break the bonds in glucose, so you can then start the process of releasing the energy until you get to your final products. All right, so again, you can see it over here with this arrow here, activation energy without the enzyme. So if you look back at this dotted line, all right, and I bring my laser pointer to here, from here to here represents the potential energy I can get out of breaking down glucose. And we had said arbitrarily it's 100 calories. But now look at this. What do you think this energy is here going all the way up this curve to this point? Just look at the distance from the starting point to up here versus really all potentially you could get out of one glucose. It's much higher, right? So let's say the activation energy is 500 calories. So you invest 500 calories just to get to this point where you can actually start to break down the bonds in glucose to release the energy, which is only 100 calories. Well, guess what? That's not a very good reaction, right? So if you're already negative 500 and you only get 100 out of the reaction, you're negative 400 calories into the situation. That was a business opportunity. Your business is going to go under and out of business quite quickly. But what enzymes do is they are that family member. They're the bank that are going to loan you the energy that's needed to start any reaction. So now this blue curve that we see here is the amount of energy. We still need some activation energy. It's still going to cost us something to get this reaction moving. All right. However, look how much lower it is now from the dotted starting line to this point here at the peak of the blue curve, where we actually can now start breaking the bonds in glucose to release its energy. This distance here, let's say maybe, is half of the 100 calories you could get out of that one glucose molecule. So now with the aid of an enzyme, all right, it costs you, let's say, 50 calories to actually be able to break down glucose at this point. You get 100 in return. Well, what's your net profit? 50 calories. You are now ahead of the game, and something happened quicker, cheaper, and easier with the aid of an enzyme. So whenever you look at a graph like this, all right, wherever your starting point is, whatever the reactants are, regardless of what's going on above it, that line, which is usually going to be in the middle of the graph, is your starting point. Anything going down below it to where the products are is the potential energy you could get out of that reaction. Well, anything then above this dotted line is going to represent the activation energy. Either the activation energy needed without an enzyme's help, which is always going to be the higher curve, or the lower curve is going to represent the activation energy, which is obviously much lower when you have the help of an enzyme. Ultimately, what's really happening, just to show you over here in these kind of little GIFs I have in the PowerPoint, if we just look at these particles floating randomly, the chances of them, right here, it's going to happen in a second. Boom, a bond forms. You can see where the square and the circle connect. That is not going to happen at a very quick, fast pace. And to get it to happen randomly would take a lot of energy. All right, so think of this up here. In order to get one bond or one reaction to occur, that's the activation energy without the help of an enzyme. But when an enzyme comes in, it helps you get things going real quick. Look, it boom, boom, grabs these two, creates the bond, releases them that fast. And then brings in another two. Boom, releases it. Enzyme will do it again. So it's just making things happen much, much quicker, reducing the energy they would need to make these things just happen randomly. All right, another way to look at it, again, you can see here, let's say this gentleman right here, all right, is trying to push this purple ball up the hill. All right, this purple ball is lactose, and I want to be able to break down lactose into its two monosaccharides of glucose and galactose. All right, here's my starting line, right, this dotted line. Here's the products. So from the dotted line to here is all the potential energy that I could get from the splitting of lactose. However, if I don't have the help of an enzyme, here's all that original activation energy I got to put into it until I can get to this point to start breaking the bonds in lactose to release energy. So just look at this amount of activation energy versus what I could get out of it. That's not a good reaction, all right? I'm not going to get anything from it. But when an enzyme comes in, It's going to make this breaking of that bond happen much quicker. Look at the activation energy needed now. Very small compared to what I'm actually going to potentially get out of that reaction. All right, so I'm not going to get 100% return. You never do that in the universe. That doesn't happen. Everything costs something, but now it's going to be much less than what it's actually going to cost without the help of an enzyme. And you can see that over here again in this GIF that I have. So see how the activation energy lowers once you have an enzyme present? So now as you lower that amount, the odds of you getting a decent amount of energy in return as you start to break these bonds, getting to the product, it makes it much more of a favorable reaction. So again, reactants to products. So anything up above it is the activation energy. Look at the amount of that, right? It's huge. But then if an enzyme comes in and helps you reduce that amount, all right, you'll see it drop again in a second, it's going to make the reaction much more favorable. So here it comes again, right? So much less of an energy input, so hopefully you get more in return. All right, some characteristics of enzymes as we move forward. Again, enzymes we're going to see are very, very specific. Generally, that means they're only going to perform one type of reaction. However, scientists have discovered over the past couple years that enzymes can actually possibly react with several different items, which we call substrates, in order to perform a reaction. Does that happen for every enzyme? No. All right, but it can for some. And we'll examine that in our next video lecture. But really, most enzymes are very specific, doing only one or maybe two different types of reactions. All right, again, as I mentioned earlier, they are reusable or recyclable. That's why I got that symbol here, meaning that once they perform the reaction, they will go back and do it again and again and again. And finally, again, each type of enzyme is only going to work within a very narrow pH and temperature range. So depending on where the enzyme is in your body, it wants to have a very specific temperature and pH. If it doesn't have that, it's not going to work properly or quickly, and it could actually break down and be destroyed if you put it out of its comfort zone. So if it wants to be at 98.6 degrees Fahrenheit, and you raise that enzyme up to 105 degrees, you have a really bad fever, the enzyme breaks down, can't do its job, and guess what? That could lead to organ failure if it happens to be within an organ in your body. And again, finishing up here, again, why are they important? Getting back to what we did in the very beginning of the year, enzymes are ultimately what is truly maintaining homeostasis within our body to keep an internal stable environment. All right, if you look at these pictures over here, maybe this is your room over here, a big cluster mess. Maybe this is your room, a super neat freak at home. But think of this, if this is your room, and mom, dad, whoever comes in says, I need you to clean your room. Well, it's going to take a considerable energy input, right, to get it like this, and it looks beautiful. But what's going to happen over the course of a day or two? It's going to start to look crazy again. And what could you do then? Put some more energy in to make it get cleaned up again. It's back and forth. These are those positive and negative feedback loops we discussed at the beginning of the year. And it's enzymes ultimately that are controlling it. So maybe you have something going on in your body that you need an organ to do. Your brain sends that message through neurons to an organ. The organ releases chemical signals. Those chemical signals will then tell enzymes that they need to either stop or start something to try to get your body to maintain homeostasis at that point. So we really have enzymes to thank for doing this very important task in our body of maintaining homeostasis. And lastly, again, enzymes can be used in a variety of different industries out there, not just in your body, but the majority of products that we use on a daily basis, pharmaceuticals, perhaps vaccines like for COVID-19, biofuels, fuels maybe that we can generate from using algae, all right, household cleaners. They all make use of enzymes on a daily basis to make things happen much quicker and efficiently for us. And we're going to do an activity that is going to, again, reinforce this idea of why we can use enzymes, not only in our body, but in industrial complexes to make things happen quicker and cost less money.